Okay, hello. I am delighted to be with you today to present this topic, paternal contribution to embryogenesis and offspring health. Let's start with an overview about this topic. The gold standard to assess fertility is by performing semen analysis. Semen analysis should be performed according to the WHO guidelines. Each couple of years, the WHO releases a new version. The latest version is published in 2021. Among others, in semen analysis, we assess sperm concentration, we assess sperm motility, and we assess the normal morphology. And we assess if there is abnormal morphology in semen, such as the presence of vacuoles inside spermatozoa. However, semen analysis alone may not predict fertility potential. Why? Because both fertilization and post-fertilization processes depend on molecular and functional sperm attributes beyond conventional assessment, such as acrosomal reaction, mitochondrial activity, genome integrity, the presence of some proteins inside the spermatozoon, such as PLC-zeta. In parallel, the activity of non-coding RNAs inside sperm, the chemical modification of the DNA, the chemical modification of the histones, these three elements are known as the sperm epigenome. All these factors are not covered by semen analysis. From this angle, the objectives of the presentation today is to examine how sperm molecular and functional parameters influence embryogenesis and offspring health. I divided the presentation into three parts. Part number 1, lab sperm handling and embryogenesis. Part number 2, lifestyle factors affecting sperm epigenome. And part 3, paternal exposure and offspring health. I will start with part 1, lab sperm handling and embryogenesis. To do this, I'll be presenting the results of our paper published in RBMO in September, entitled Sperm Vacuolization and Mitochondrial Activity in Raw Semen versus Prepared Semen, Impact on Embryological Outcomes. Introduction. Sperm preparation techniques isolate progressive motile sperm from seminal plasma, white blood cells, and debris. So in IVF, we use several techniques to select progressive motile sperm, such as swim up technique relies on the ability of motile spermatozoa to swim out from the semen up to the sperm culture medium, or using a density gradient technique, which selects morphologically normal motile spermatozoa based on their specific density. After sperm preparation, motile morphologically normal spermatozoa with intact DNA and active mitochondria are usually selected. However, under certain circumstances, sperm damage may happen due to centrifugation, to oxidative stress during handling. Thus, sperm characteristics following preparation may differ from those in raw semen. And the impact of these changes on embryo development remains to be elucidated. Therefore, the objective of the study was to evaluate whether sperm vacuoles, DNA fragmentation, acrosomal status, mitochondrial activity better predict fertilization and blastulation rates after preparation than in raw semen. How did we proceed to answer this question? We've enrolled infertile couples undergoing ICSI cycles at Al Hadi IVF Center, Beirut, Lebanon, between January and December 2024. We excluded couples undergoing ICSI using frozen gametes, testicular and/or embryo biopsy, female partners with a body mass index more than 30, female partners with an advanced maternal age, or female partners having less than 3 oocytes collected. We ended up with 90 couples. On the day of oocyte pickup, each couple provided a semen sample. The semen sample was divided into two portions, raw semen and another portion was prepared using density gradient or swim up. In parallel, mature oocytes were collected from the women, and each mature oocyte were injected by a spermatozoa. From the leftovers of the raw semen and the post-preparation, we analyzed sperm vacuoles according to Kruger-restricted criteria, sperm mitochondrial activity using DAP staining, sperm DNA fragmentation using STD technique, and sperm acrosomal reaction using triple staining technique. In addition, we collected data from the lab regarding the fertilization rate and the blastulation rates. What are the results? So as you can see on in the red line, it's fertilization rate. On the y-axis, you can see all the sperm parameters that we have assessed. Below, we have the basic parameters, then DNA fragmentation, acrosomal status, and mitochondrial activity. Interestingly, a significant negative correlation was found between vacuoles post-preparation and fertilization rate, with a p-value of 0.008. We tried to quantify this association between vacuoles and fertilization. We did a linear regression, and we found that fertilization rate was 79.78% when no vacuoles were present post-preparation. Each 1% increase in vacuoles decreased fertilization rate by 1.39%, with a p-value of 0.001. So there's a predictive relationship between vacuolated spermatozoa post-preparation and fertilization rate. Now regarding blastulation rate. On the red line, you can see blastulation rate. On the y-axis, you can see the sperm parameters from the basic one to DNA fragmentation, acrosomal reaction, and mitochondrial activity. Interestingly, we found a significant positive correlation between spermatozoa with active mitochondria and blastulation rate, with a p-value of 0.03. We tried to quantify this association using linear regression. We found that blastulation rate was 21% when no sperm with active mitochondria were found in the semen. Each 1% increase in mitochondrial activity in spermatozoa and raw semen increased blastulation rate by 0.37%, with a p-value of 0.03. So as a conclusion of this study, the percentage of post-preparation spermatozoa exhibiting vacuoles may predict fertilization rate. In parallel, the percentage of spermatozoa with active mitochondria in raw semen may predict blastulation rate. It's worth mentioning, vacuoles were less prevalent when the swim-up method was applied for sperm preparation. Let's move to the second part of this presentation, lifestyle factors affecting sperm epigenome. Usually a cell, under certain conditions, can activate its genes by increasing the amount of RNAs and increasing the amount of proteins, or can undergo gene silencing and decrease the transcription and translation of RNAs and proteins respectively. How a cell can regulate the amount of RNAs? It can use the epigenetic control. What is an epigenetic control? It's adding chemical modification on the DNA, such as DNA methylation and DNA hydroxymethylation, by adding chemical modification to the histones, or by the activity of non-coding RNAs. These three elements are known as the epigenome. This epigenome controlling gene expression can be influenced by metabolism, by toxins, by active oxygen species. So exposure to certain environment can modify DNA methylation, can modify histone modifications, and can modify the activity of non-coding RNAs. These modifications may predispose the patient or the person to diseases or to changes in the behavior. The male reproductive system is known that if it was exposed to toxins, endocrine disruptors, smoking, or obesity, may alter the sperm epigenome, and these alterations in sperm epigenome may alter the gene expression in embryos, predisposing this embryo to diseases later in life. From this angle, We conducted a study which was published in Journal of Translational Medicine last August, entitled Iron Biomarkers in Men with Infertility are Associated with Sperm DNA Hydroxymethylation and Cumulative Live Birth Rates, a prospective study. Why iron? Iron is very important for body metabolism, for oxygen storage, oxygen transport, and as well for spermatogenesis. Once it's absorbed by the intestine, iron binds to plasma transferrin and circulates in the body. And once inside the cell, iron is stored inside ferritin. Of particular interest, inside the testicle, Sertoli cell may produce transferrin to ensure an optimal amount of iron to the germ cells. Clinically, we can measure the iron status by measuring ferritin, serum iron, transferrin, and by measuring the potential of transferrin to bind to iron, such as UIBC and TIBC. Maintaining iron homeostasis is essential to prevent the generation of oxidative stress, because iron deficiency and excess free iron both can lead to oxidative stress, and this OS may impair the sperm quality. In addition, iron may activate and regulate the activity of a crucial enzyme called TET. TET stands for 10-11 translocation enzyme. It's an iron-dependent enzyme, and it's a metabolic-dependent enzyme. What is the function of TET? TET may help in the transformation of 5-methylcytosine in the DNA to 5-hydroxymethylcytosine. So TET is involved in controlling gene expression. This enzyme, which is an iron-dependent enzyme called TET, is expressed in testicles and in all the line of spermatogenesis. However, the influence of iron biomarkers on sperm DNA hydroxymethylation has not yet been fully investigated. Therefore, the aim of the study was to assess whether iron biomarkers in men are associated with the global DNA hydroxymethylation in spermatozoa. How did we proceed? We've enrolled infertile men attending Al-Hadi lab in Beirut for infertility treatment. We excluded all men using frozen semen, testicular biopsy, or underwent any previous cytotoxic treatment such as chemotherapy. So we included 60 men. Each man provided a blood sample and a semen sample. A fraction of semen sample along with a fraction of the blood were used to assess iron biomarkers. The other fraction of the semen was subjected to density gradient to select motile spermatozoa. DNA was extracted from motile spermatozoa, and the extracted DNA was subjected to ELISA to assess the global DNA hydroxymethylation. The results. Interestingly, serum iron and seminal fluid iron both were positively correlated with global DNA hydroxymethylation in spermatozoa. In addition, seminal fluid UIBC levels were positively correlated to the global DNA hydroxymethylation with a p-value of 0.04. Moreover, serum TIBC and seminal fluid TIBC both were positively correlated with global DNA hydroxymethylation. So as a list, serum iron and serum TIBC, in addition to seminal fluid iron, seminal fluid UIBC, seminal fluid TIBC, they were associated with global DNA hydroxymethylation. Then we wanted to check which one from this list is the most important for global DNA hydroxymethylation. We did a multivariate analysis, and we found that serum TIBC had a statistically significant positive association with sperm DNA hydroxymethylation with a p-value of 0.02. How can we explain this finding? When iron bioavailability is low inside the body, the concentration of transferrin and the binding capacity of transferrin will increase to ensure the optimal amount of iron to the cells. This increase in iron inside the cell may activate the TET enzyme, leading to an increase in hydroxymethylation. However, what is the impact of this increase in hydroxymethylation of the offspring health? This is a big question to answer in further studies. And based on this question, we will open the third part of this presentation, which is paternal exposure and offspring health. Several meta-analyses and studies in humans and animals showed that paternal exposure to certain environments, such as obesity, smoking, alcohol, may alter the sperm epigenome. I gave you an example on iron from the previous study. And these alterations may be associated with the health of the offspring. In this context, we've published an article with my students in andrology entitled Preconceptional paternal obesity may increase the risk of congenital urogenital anomalies in offspring, a case control study. What did we do? We've included newborns, 91, with congenital urogenital malformations, and 88 newborns without any malformation. Then we excluded all the parents with maternal obesity, infections during pregnancy, having chronic diseases, underwent assisted reproductive technology, maternal substance abuse, etc. And we asked the parents about the obesity of the father at the time of conception. And we found that compared to normal weight fathers, overweight fathers displayed a heightened risk of offspring congenital malformation with an odds ratio of 4.44. So paternal obesity may increase 4 times the risk of having congenital urogenital malformations in the offspring. We've conducted another study, also published in Andrology, entitled Paternal Periconceptional Physical Activity and the Risk of Congenital Heart Disease in Offspring, a case control study. In this study, we included babies with congenital heart disease, 128 cases, and 151 controls without congenital heart disease. And we asked about the paternal exposure at the time of conception. What did we ask? We asked about the socio-demographic characteristics, the medical history, and we assessed physical activity using a global physical activity questionnaire, which is a questionnaire validated by WHO, made up of 16 questions with 4 dimensions, related to activity at work, travel-related activity, sedentary activity, etc. And we found that paternal involvement in physical activity was associated with a reduced risk of congenital heart disease development in offspring by 46.9%, and a p-value of 0.02. As a general conclusion, the presented studies indicate that spermatozoa may contribute to embryogenesis and offspring health through functional and molecular characteristics beyond analysis. Further research is required to better characterize these associations and to clarify their relevance for reproductive outcomes in fertile and infertile population. Thank you for listening. Dr. Fadi, the floor is yours.
Yeah, thank you, Dr. George, for this insightful presentation.
Thank you.
So, as we all know, Dr. George, he's an accomplished author of high-impact publication in international peer-reviewed journals with expertise in reproductive biology and embryology. And you all know he has a PhD in clinical and therapeutic research from Côte d'Azur University, France, and he's an assistant professor at the American University of Beirut. Thank you for this insightful presentation, and I'll move on to my talk.
While preparing this presentation, I'll present you Dr. Fadi Sheh, a scientific manager and the lead clinical embryologist at CIDRA Medical and Research Center, SHA-certified senior embryologist. Dr. Fadi is also the chair of the Middle East Fertility Society, special interest group in embryology, He is an associate editor of the Journal of Translational Medicine and Middle East Fertility Society. He holds a PhD in the field of transcriptomics and reproductive epigenetics from the University of Côte d'Azur, France. Dr. Fadi, the floor is yours.
Thank you. Can you hear me? Can you see my screen also?
Yes, we can.
Yeah. Thank you, Dr. George, and thank you for the organizing committee for this kind invitation. And thank you for the audience from the Arab world and mainly from Iraq for joining us for this late hour. So you're eager to learn more, and we're eager to hear your question at the end of the session. So in the next 20 minutes, I will present for you insights into pre- and post-processing sperm factors in assisted reproductive technologies. So you will be, in the next 20 minutes, at the embryology lab gate. Nothing to disclose. So learning objectives, by the end of this session, you will be able to understand the significance of sperm quality in the context of ART success rates, identify how collection methods, sample handling influence the quality of sperm received by the ART lab. So the first key question raised is why pre- and post-processing of semen samples matters in ART. So why it matters? Why we need to care about the pre- and post-processing of the samples. So why conventional semen parameters may not always predict ART success. Conflicting data, so as you know, doesn't impact the success. However, data from oocyte donation models, which provide a clearer assessment of the sole impact of sperm quality. These studies suggest that poor sperm morphology and motility are associated with poor laboratory outcomes and poorer clinical outcomes, reinforcing the importance of pre- and post-processing evaluation in optimizing ART outcomes. Another advanced sperm parameters in sperm is sperm DNA fragmentation, which has been associated with poor clinical outcomes, particularly in conventional IVF. Also, evidence from oocyte donation programs, which will help us to isolate the sperm factor, suggests a negative correlation between high sperm DNA fragmentation or high DNA fragmentation index and reduced fertilization rates, impaired embryo development, and lower embryo quality. So these findings highlight the critical role of sperm DNA integrity in both laboratory and clinical ART success. So in the ART laboratory, the andrology section, is responsible for handling and processing semen to optimize its potential for fertilizing oocytes and supporting embryo development. However, processing techniques can either enhance or in some cases further compromise sample quality. So one example is sperm freezing, where a growing body of evidence indicates that freeze-thaw process reduces progressive motility and increases sperm DNA fragmentation and potentially affecting clinical outcomes. Another critical aspect is sperm preparation, sperm processing, as different processing methods can have varying effects on sperm parameters and laboratory outcomes, underscoring the need for careful selection of techniques to optimize ART success. So building on the importance of pre- and post-processing in ART, we will now address key considerations in sperm pre-processing. So exploring how different collection and handling practices influence fertilization potential, embryo development, and overall clinical outcomes. So the collection method is a critical factor influencing semen quality. the WHO recommends a sexual abstinence period for semen ejaculates of 2 to 7 days. So a guideline based on standardized reference values and examination protocols. This recommendation aligns with the epididymal sperm reserve, which corresponds to 1 to 2 days of testicular sperm production, with maturation of the sperm taking less than 2 days. However, the duration of abstinence is a key variable affecting sperm quality. So clinical studies indicate that longer abstinence is associated with reduced total and progressive motility and increased tail defects, particularly in cases with pre-existing sperm abnormality. Conversely, meta-analysis data suggests that shorter abstinence periods can improve sperm progressive motility and reduce sperm DNA fragmentation index, even in samples with abnormal sperm parameters. So these findings underscore the need to carefully consider abstinence duration as part of semen collection protocols to optimize ART outcomes. So basic biology data indicate that prolonged abstinence more than 7 days is associated with reduced sperm motility and abnormal morphology due to sperm senescence. This phenomena is primarily attributed to an increase in reactive oxygen species, which accumulate when sperm remains stored in the epididymis for extended periods. Another consideration are some pre-collection practices. While the WHO recommends urination, passing urine before semen collection to reduce microbial contamination, evidence suggests that this may be ineffective due to discrepancies between urine and semen cultures. It is crucial also to consider the impact of collection materials on semen quality. The use of natural and synthetic lubricants has been associated with negative effects on sperm motility and chromatin integrity. However, some isotonic lubricants appear to have minimal adverse impact on sperm parameters, making it a potential alternative for use in a fertility setting. So over-the-counter non-spermicidal condoms have been shown to be highly toxic to sperm total and progressive motility. Additionally, the use of sterile non-toxic containers that do not alter motility characteristics or introduce embryo toxicity is recommended. It is also critical to emphasize and document ejaculate loss, as it may impact sperm quality and treatment outcomes. Research on split ejaculates has demonstrated the superiority of the first ejaculate portion, which contains higher sperm count and progressive motility. Another important aspect to consider is sample handling and transport, as these factors can significantly impact sperm quality. So ejaculate to analysis delay has been shown to negatively impact sperm motion and motility parameters after 1 to 1.5 hours, reinforcing the WHO manual recommendation to process samples within half an hour to 1 hour. As for temperature, sperm motility appears to remain relatively stable when maintained at or near body temperature for up to 2 hours. Interestingly, data from the COVID-19 period indicate no significant association between collection location, home versus clinic, and key sperm parameters, including motility, total motile count, embryological and clinical outcomes. In addition to the challenges associated with ejaculate handling and transport, surgically retrieved and recovered sperm samples from the testicle present another complexity. When a suspension is dominated by erythrocytes, by RBCs, sperm identification and retrieval can become more difficult, increasing the time required for selection inside the embryology lab. So to mitigate this issue, erythrocyte-lysing buffer is often used. However, its effect on sperm function remains debated, with concerns about potential negative impacts. This underscores the importance of retrieving testicular tissues with minimal blood contamination to ensure sperm recovery while preserving function and integrity. Regarding the transport medium, which medium we transport surgically retrieved testicular tubules, normal saline, because you know some urologists, they might have suggest using irrespective medium. So just to emphasize that normal saline is not ideal for sperm preservation, as it lacks essential nutrients, antioxidants, and buffering agents required to maintain sperm viability over extended periods. Early studies indicate that normal saline significantly reduces sperm motility, likely due to its lower pH compared to sperm pH, which is 8.2. Since pH plays a fundamental role in sperm function, impacting capacitation and the acrosome reaction, using an appropriate medium is crucial. So it is recommended to use commercial sperm washing capacitation or culture medium, as these solutions mimic in vivo physiological conditions, promote higher motility, and contains nutrients, antioxidants, and HEPAs or MOPs, the buffers for maintaining sperm function and pH stabilization. Regarding temperature, for surgically retrieved samples, media should be pre-equilibrated at 37 degrees to maintain pH stability and support capacitation. So for short transport, for short transport, room temperature is acceptable. However, experience from tissue banks in a network of US clinics in the United States suggests that transporting tissue on ice 4 degrees does not compromise tissue morphology or apoptosis rates, demonstrating an alternative approach for preserving sample integrity. The ideal processing time for ICSI or cryopreservation is generally within 24 hours to use surgically retrieved sperms, as supported by bovine models, which show that this time frame, 24 hours, helps maintain sperm morphology, viability, apoptosis rates, and testis-related gene expression. Interestingly, a 24-hour incubation of testicular spermatozoa has been found to enhance motility and fertilization outcomes, thought same-day processing within a few hours remain the widely accepted practice. Additionally, accurate tube labeling is critical in IVF labs. While labeling errors are rare, they can have serious consequences, highlighting the importance of stringent quality control measures to ensure patient safety and procedural accuracy. So now we'll move on to post-processing considerations. So we discussed the pre-processing before getting the sample inside the lab. Now we'll move on to the post-processing consideration. Sperm processing and separation. So first, in the embryology lab, we assess the count and the motility. So we do a basic semen analysis. And the WHO emphasizes the need for a proper liquefaction. So performing semen analysis prior to complete liquefaction can compromise the accuracy of the results, particularly concerning its sperm motility and concentration. As for the analysis, routine semen analysis at 37 degrees is required for standardization of analysis and temperature fluctuation control, because temperature fluctuation has been earlier showing that it affects motility. So that's why in the embryology lab, we have microscopes with an integral heated stage and pre-warmed slides. Also, a recent study showed that delaying covering the chamber, the counting chamber where we assess the motility, affects the accuracy of the results. It will overestimate the sperm concentration and motility. Here we see, like this analytical study, they compare the timing between if you're immediate or if you took time to cover the chamber, counting chamber. Now we'll move on to the ejaculation to analysis timing. So studies have shown that ejaculation to analysis delay negatively influence sperm motion parameters after 1 to 1 hour and a half. That's why WHO recommends processing within maximum 1 hour. And microscopic examination should be done within 1 minute, because any drifts will impact the result. As for morphology, WHO recommends staining techniques, Papa Nicolau or quick diff. However, errors or artifacts might arise due to variability in sperm dimension from different stains. Inaccuracies due to subjectivity also and interpretation. That's why in the embryology lab we have QCs, we have external proficiency testing where we, inside the lab, we have programs where we do the assessment of the morphology and we compare them with other external labs or with other regulatory, with other boards like CAP, like NECWAS in the UK. And training also is required because inaccuracies will impact interpretation. Now let's move on to the processing in the lab, to the wash, to the preparation of the sample. Early studies have shown that bacterial contamination was highly decreased by gradient centrifugation and almost abolished when strict aseptic techniques were used. So with changing the sterile Pasteur pipettes and changing the tubes prior to washing procedures, as you see here in the bottom histogram. Also, the time of centrifugation is also critical because time of centrifugation, if it's increased, might induce ROS formation in the semen. Now for the processing, for the washing methods, you know, conventional methods in the lab, we have the conventional basic ones, density gradient, swim up. So no difference in pregnancy rate for density gradient versus swim up in IUI cycles, which is in normosospermic samples. That's the data from Cochrane database, which is level 1 evidence. However, compelling evidence, and here an interesting prospective study showed that semen swim up is more effective. This study was conducted by Dr. George, more effective than density gradient in selecting better spermatozoa in terms of DNA integrity and reactive oxygen species levels. However, the only evidence found in the literature showed that the clinical outcomes between density gradient and swim up is similar. However, density gradient, it yields sperm population with high motility and better morphology. Now let's move on to not the ejaculate, to the surgically retrieved testicular tubules. So aspirate or dissected tubules, lysates or homogenate, separated on density gradient, can be separated later on density gradient or simple wash and direct examination. So those seminiferous tubules can be mechanically minced, macerated, and shredded or enzymatically digested. So you can use mincing with fine needles, scissors, and other mechanical methods, and optional addition of collagenase and erythrocyte lysis buffer. So collagenase type 4 and elastase for 1 hour. So in some practices, they will incubate lysates with collagenase for 1 hour at 37 degrees, which resulted in higher yield of spermatozoa because spermatozoa were hidden within the testicular tubules. So studies have shown that also sperm suspension can be exposed to erythrocyte lysing buffer for 5 to 10 minutes to facilitate the search during ICSI. However, a recent study showed that erythrocyte lysing buffer we should limit using it because it decreased motility, it impacted viability, and it increased DNA fragmentation. Now the mincing of the testicular tubules, effective mincing is important because to ensure all seminiferous tubules are disrupted and intratubular cells are freed into the suspension. Because if we didn't mince well, residual tissue can block the migration of spermatogenic cells through the gradient layers. And then we will lose the sperm cells because they will be stuck and blocked within the gradient layers. Okay, so now to take home messages. So what happens inside the lab depends heavily on what happens before the gate of the lab. The second take home message, every step from sample collection to sperm injection for inseminating the oocytes is part of a continuum, and it's not an isolated event. Urologists and embryologists and andrologists are partners and are not separate silos. And the better the starting material, the better the embryologist can do their job. And at the end, at the end, I encourage you to attend the Middle East Fertility Society, which will be happening. Let me share my screen, which will be happening on the 21st to the 23rd of November in Jeddah, Saudi Arabia. So I encourage every one of you interested to, and we will be more than happy seeing you in the meeting. 1 second, let me share. Can you see my slide? So I encourage you to register and to see you there. We can catch up there where you can see many innovations and new insights into the field in Jeddah, Saudi Arabia at the end of November 2026. And thank you again for the invitation.
Thank you, Dr. Fadi, for this amazing presentation. Very useful.
Thank you.
Mr. Anas, do we have some questions from the audience?
Yeah.
Okay,
Are there
thank
any
you.
questions? I will, Dr. George, I can ask you a question. So you presented an interesting paper, sperm vacuolization and the impact and the association with clinical outcome, which was published in a prestigious journal. So, so what's your insights regarding the old practice using IMSI? You know, IMSI has been a hype maybe in early 2010, 2013, and most of the labs, they got IMSI because vacuoles, it was found associated with DNA fragmentation. And later, the compelling evidence showed that it's not it's just an artifact or the anatomy of the sperm. So what's your comment based on your data? And
Yes.
is there any place of IMSI now in 2026 in the lab? So, okay.
Okay, I'll get inspired from your presentation, pre and post. I will tell you pre-ICSI and post-ICSI. So in this paper, I presented a part of it. So we analyzed vacuoles before sperm preparation, after sperm preparation, and after ICSI, the outcomes. So using inappropriate techniques to prepare spermatozoa may induce a stress in the whole sample. So what we showed that if the wall sample is under stress and the percent of vacuole will increase, the percentage of fertilization will decrease. So, and this is on one thing. So it's not MC and selecting particularly sperm without vacuoles, it's the population of sperm under stress. Two, we found in this paper, we performed a mediation analysis. What is mediation analysis? If vacuoles is a mediator between factor X and the fertilization. So vacuoles are also mediators. They can mediate the effect of DNA fragmentation on embryo development. They can mediate the effect of acrosomal action on embryo development. So inducing vacuolization and imposing stress on sperm during preparation is the thing to consider. So it's similar somehow to your presentation.
Yeah, thank you. Thank you, interesting. Do you have any interactions from the audience?
I think they can email us later on in case they have any question, and we're more than happy to answer.
Yeah, it's a great idea. So, Anas, we can share also our email addresses, and if there's anyone can have any question, or also you can reach us on social media platforms, on LinkedIn. You can on LinkedIn just send us a message if there's any question related to the topic.
Thank you, Dr. Fadi. Thank you for your time.
Thank you again. And thank you also for the audience staying for the attending during a weekend a scientific session. Thank you.